Let's start with something incredibly simple, something you might even overlook at your local grocery store: a nut. But not just any nut. This one comes from the heart of the Amazon rainforest, the Brazil nut. And hidden inside that unassuming shell lies a nutrient that's become the subject of dozens of studies across the globe. A micronutrient that your body doesn't need in large amounts, but one that carries powerful, far-reaching effects across multiple biological systems: selenium. Today, I walk you through the astounding benefits of selenium found in Brazil nuts, not just from a nutritional perspective, but from a cellular, hormonal, cardiovascular, and even neurological standpoint. We often talk about superfoods, but rarely do we talk about the super mineral that selenium truly is. And in a world where chronic disease is now our primary health crisis, where oxidative stress and inflammation play pivotal roles in everything from heart disease to cancer to cognitive decline, the simple process not may just be one of the most overlooked tools in your health arsenal. Selenium is a trace mineral that plays an essential role in the human body, particularly due to its involvement in antioxidant defense systems. While often overlooked in daily dietary considerations, selenium is a critical cofactor for the enzyme glutathione peroxidase, one of the most powerful antioxidants in the body. This enzyme is responsible for reducing harmful reactive oxygen species, particularly hydrogen peroxide, into water, thereby preventing oxidative damage within cells. Hydrogen peroxide is a natural byproduct of cellular metabolism, but when it accumulates in excess, it can generate free radicals that attack lipids and DNA. This process, known as oxidative stress, is a major contributor to aging, inflammation, and chronic diseases. The body's ability to neutralize oxidative stress depends heavily on the presence of antioxidants, and glutathione peroxidase is at the forefront of that defense mechanism. Without adequate selenium, the activity of this enzyme drops significantly, leaving cells more vulnerable to oxidative injury. The role of glutathione peroxidase is not limited to a single area of the body. It operates across various tissues, including the liver, brain, heart, kidneys, and immune cells. In the liver, it helps detoxify peroxides that are formed as a result of drug metabolism or exposure to environmental toxins. In the cardiovascular system, it plays a protective role by preventing the oxidation of LDL cholesterol, a key step in the development of atherosclerosis. In the brain, where neurons are particularly sensitive to oxidative damage due to their high metabolic demand and limited regeneration capacity, glutathione peroxidase serves as a crucial line of defense against neurodegeneration. All of this hinges on selenium being present in sufficient amounts, not in excess, but in the precise microgram quantities the body requires daily. This is why selenium is referred to as a micronutrient. It is needed in small amounts, yet its absence can result in significant cellular dysfunction. The antioxidant defense provided by glutathione peroxidase is particularly vital under conditions of physiological stress, such as infection, inflammation, or exposure to pollutants. During these times, the generation of reactive oxygen species increases, and so does the demand for antioxidant capacity. If selenium stores are depleted or insufficient, the body's ability to upregulate glutathione peroxidase is compromised, leaving it vulnerable to unchecked oxidative stress. This imbalance can lead to damage of cell membranes, mitochondrial dysfunction, and the triggering of chronic inflammatory pathways. These are the underlying mechanisms behind many age-related diseases, staying in heart disease, cancer, diabetes, and neurodegenerative disorders. In this context, selenium acts almost like a nutritional that determines whether the body moves cellular resilience or cellular damage. Brazil nuts are among the richest natural sources of selenium, with a single nut providing more than the recommended daily intake for most individuals. What makes this food unique is its ability to deliver selenium in its organic form, known as selenium-methionine, which is highly bioavailable and efficiently utilized by the body. Unlike synthetic supplements that may vary in absorption and safety, selenium from Brazil nuts comes naturally bound within a whole food matrix, which improves its biological effects and reduces the risk of toxicity when consumed in appropriate quantities. The regular consumption of even one Brazil nut per day can significantly increase serum selenium levels and support the optimal function of glutathione peroxidase. Moreover, research has demonstrated that selenium status in humans correlates closely with the activity of glutathione peroxidase in plasma and red blood cells. Individuals with higher selenium levels tend to exhibit greater enzymatic activity, which translates into better oxidative balance and lower markers of systemic inflammation. This is not merely a theoretical connection. It has been observed in clinical trials and population studies across different demographics. In areas of the world where soil selenium content is low, such as parts of China and New Zealand, people tend to exhibit lower glutathione peroxidase activity and higher rates of oxidative stress-related conditions. This association underscores the vital role selenium plays not just in isolated cellular reactions, but in the broader context of public health. When selenium is integrated into the structure of glutathione peroxidase, it becomes indispensable to that enzyme's catalytic function. Unlike vitamins or other cofactors that can be substituted, the absence of selenium directly results in the failure of glutathione peroxidase to perform. That makes it a rate-limiting factor in the body's ability to control oxidative stress. This is particularly important in high-energy organs like the heart and brain, where oxidative injury can rapidly lead to irreversible damage. For example, during a heart attack or stroke, the burst of oxidative stress can be so severe that antioxidant defenses are overwhelmed unless they are already primed and supported by adequate selenium levels. This preparatory role of selenium, quietly maintaining the integrity of antioxidant enzymes, is a powerful form of cellular insurance against both acute and chronic damage through its essential role in glutathione peroxidase. Selenium enables the body to neutralize free radicals and maintain redox balance at the cellular level. This balance is critical for preserving mitochondrial function, preventing DNA damage, and inhibiting inflammatory signaling cascades. Selenium plays a fundamental role in thyroid health and hormonal balance, serving as a cofactor for several enzymes that are essential to thyroid hormone metabolism. The thyroid gland contains more selenium per gram of tissue than any other organ in the human body, staying away from processed, which highlights just how important this trace mineral is for proper thyroid function. One of selenium's key contributions is its involvement in the activity of isothreonine dehydrogenases, a group of selenium-dependent enzymes responsible for the activation and deactivation of thyroid hormones. These enzymes convert thyroxine T4-2, the inactive storage form of thyroid hormone, into triiodothyronine NT3, the biologically active hormone that influences metabolism, energy production, temperature regulation, and overall cellular function. Without sufficient selenium, the efficiency of this conversion drops, leading to a reduction in available T3 even if total thyroid hormone levels appear normal on lab tests. In addition to its role in hormone conversion, selenium also protects the thyroid gland from oxidative damage. The process of producing thyroid hormones requires large amounts of hydrogen peroxide, which, if left unchecked, can damage the delicate tissues of the thyroid itself. Here again, selenium-dependent enzymes such as glutathione peroxidase play a crucial protective role by neutralizing hydrogen peroxide before it causes cellular harm. The thyroid is uniquely vulnerable because it constantly operates in a pro-oxidative environment due to its reliance on iodine oxidation to produce hormones. This makes antioxidant protection, especially from selenium-dependent enzymes, absolutely essential to maintaining thyroid integrity over time. Selenium's role goes even further when considering its involvement in autoimmune thyroid conditions, particularly Hashimoto's thyroiditis and Graves' disease. Both conditions involve the immune system mistakenly attacking the thyroid gland, leading to chronic inflammation and either underproduction or overproduction of thyroid hormones. Research has shown that low selenium levels are associated with a higher prevalence and severity of autoimmune thyroid diseases. Supplementation with selenium has been found to reduce levels of thyroid peroxidase antibodies, TPOAB, which are markers of autoimmune attack against the thyroid. In individuals with Hashimoto's, selenium supplementation has led to a measurable decrease in these antibodies, suggesting that it helps modulate the immune response and reduce inflammatory damage within the thyroid tissue. Hormonal balance extends beyond the thyroid, and selenium's indirect influence on other endocrine pathways is becoming increasingly recognized. For example, the adrenal glands, which produce cortisol and other stress-related hormones, are also sensitive to oxidative stress. Selenium's antioxidant role helps preserve adrenal function, ensuring that the body's response to stress remains balanced and does not become dysregulated. This interplay between selenium, oxidative stress, and endocrine regulation is part of a larger network where micronutrient status can significantly influence hormonal signaling throughout the body. Selenium also interacts with insulin signaling and glucose metabolism. While not a direct component of insulin production, selenium's support of antioxidant systems helps prevent oxidative damage to pancreatic beta cells, which are responsible for insulin secretion. Oxidative stress in these cells is a known contributor to the development of insulin resistance and type 2 diabetes. By maintaining redox balance, selenium indirectly supports healthier glucose regulation and insulin sensitivity. This connection illustrates the broader impact of selenium on hormonal pathways outside the traditional thyroid context. The reproductive system also relies on selenium for hormonal regulation. In men, selenium is essential for testosterone synthesis and healthy sperm production. It is concentrated in the testes and plays a role in the structural integrity of sperm cells, contributing to fertility. In women, selenium helps regulate estrogen metabolism and supports the hormonal changes that occur during the menstrual cycle, pregnancy, and menopause. Its anti-inflammatory and antioxidant properties are especially valuable during periods of hormonal fluctuation, where oxidative stress may increase and hormone balance becomes more delicate. Inadequate selenium intake may contribute to symptoms that mimic hormonal imbalance, such as fatigue, weight gain, sensitivity to cold, depression, and hair thinning. These symptoms are commonly associated with hypothyroidism, but can also stem from a lack of T3 conversion due to low selenium levels. Since many conventional lab tests do not differentiate between T4 and T3 or measure selenium status directly, individuals may experience symptoms of deficiency even with normal thyroid lab values. This is one reason why dietary sources of selenium like Brazil nuts, offer a practical and effective way to support hormone health without the need for supplementation. In many cases, Brazil nuts provide selenium in a natural, bioavailable form that supports the body's enzymatic pathways involved in hormone production, regulation, and detoxification. Just one nut can deliver a significant amount of selenium, making it a highly efficient food-based solution for individuals looking to support their thyroid and hormonal systems. The combination of selenium's antioxidant capacity, enzymatic support, and immune modulation forms a powerful triad that directly influences hormonal balance across multiple systems. Selenium plays a vital role in supporting cardiovascular health, not by lowering cholesterol directly, but by addressing the oxidative and inflammatory processes that underlie the development of heart disease. One of the most important mechanisms by which selenium supports the heart is through its involvement in preventing the oxidation of low-density lipoprotein, or LDL cholesterol. While LDL is often labeled as bad cholesterol, it only becomes harmful when it is oxidized. Oxidized LDL contributes to the formation of atherosclerotic plaques, which narrow arteries and reduce blood flow, ultimately increasing the risk of heart attacks and strokes. The enzymes that prevent this oxidation, particularly glutathione peroxidase and thioredoxin reductase, are selenium-dependent. These enzymes neutralize hydrogen peroxide and lipid peroxides that otherwise promote the transformation of LDL into its oxidized damaging form. When selenium is present in adequate amounts, the antioxidant defense system remains active and effective, especially within the endothelium, the inner lining of blood vessels. The endothelium is a dynamic organ that regulates vascular tone, blood flow, and immune cell activity. Oxidative stress damages this delicate lining, triggering inflammation and reducing the production of nitric oxide, a key molecule that allows blood vessels to relax and maintain healthy blood pressure. Selenium, through its antioxidant function, protects the endothelium and preserves nitric oxide bioavailability. This support is essential for maintaining vascular elasticity, preventing hypertension, and reducing the progression of vascular stiffness with age. Selenium also influences systemic inflammation by modulating the expression of pro-inflammatory cytokines and reducing levels of C-reactive protein (CRP), an inflammatory marker commonly elevated in cardiovascular disease. Studies have demonstrated that individuals with low selenium levels tend to have higher CRP concentrations and greater risk for coronary artery disease. By restoring selenium status, inflammation is reduced at the molecular level, which may slow the progression of arterial plaque buildup. This anti-inflammatory effect is not limited to the arteries. It also benefits the entire circulatory system, lowering the risk of clot formation and improving overall circulatory efficiency. In addition to these protective effects, selenium influences cellular energy production within heart muscle cells. The heart is an organ with extremely high energy demands and a dense concentration of mitochondria. Mitochondria are not only the powerhouses of the cell, but also major sources of reactive oxygen species. Selenium supports mitochondrial integrity by maintaining redox balance and protecting these energy-producing structures from oxidative degradation. A deficiency in selenium compromises mitochondrial function, leading to reduced ATP production and potentially weakening the heart's ability to contract efficiently. This is particularly concerning in individuals with existing heart failure or cardiomyopathy, where selenium's role in energy metabolism becomes even more critical. One of the clearest examples of selenium's importance in heart health is seen in Keshan disease, a form of cardiomyopathy first identified in regions of China where soil selenium levels are extremely low. People in these regions develop severe heart enlargement and dysfunction due to selenium deficiency. When selenium was supplemented in the population, the incidence of Keshan disease dropped dramatically. This condition illustrates how essential selenium is for cardiac muscle structure and function. Even outside of traditional risk factors like diet, exercise, and genetics, selenium also plays a role in lipid metabolism by influencing the activity of enzymes involved in fatty acid oxidation. When fatty acids are not properly metabolized, they can accumulate and contribute to lipid abnormalities that increase cardiovascular risk. Selenium's support of metabolic enzymes helps ensure that fats are efficiently utilized for energy rather than stored inappropriately or deposited in arteries. Furthermore, selenium's antioxidant activity helps stabilize cell membranes, including those of red blood cells and endothelial cells, contributing to better circulation and tissue oxygenation. Recent studies have begun to investigate the synergistic effects of selenium with other micronutrients such as coenzyme Q10 in supporting heart health. In some clinical trials, elderly individuals who received both selenium and coenzyme Q10 supplements experienced significantly reduced cardiovascular mortality compared to those who received a placebo. These results suggest that selenium's effects may be amplified when combined with other nutrients that support mitochondrial and antioxidant function, offering a more comprehensive protective strategy against age-related cardiovascular decline. Brazil nuts, as a natural source of selenium, provide an easy and accessible way to incorporate this mineral into the diet for cardiovascular support. Their high selenium content means that consuming just 1 or 2 nuts a day can make a meaningful difference in selenium status. Unlike supplements, which can sometimes lead to excessive intake, whole foods like Brazil nuts also offer fiber, healthy fats, and other phytonutrients that support heart health. This makes them an ideal addition to a heart-conscious diet that emphasizes nutrient density and antioxidant support. The heart is not just a mechanical pump, it is a dynamic, metabolically active organ that requires constant support to maintain its function throughout life. Selenium contributes to this support through a multifaceted approach that includes oxidative protection, inflammation reduction, mitochondrial support, and lipid regulation. Selenium is a cornerstone in the function of the immune system, acting as a vital nutrient that enhances both innate and adaptive immunity. Its presence is required for the proliferation and function of several immune cell types, including T cells, natural killer cells, macrophages, and neutrophils. These cells play frontline roles in identifying and destroying pathogens, clearing infected cells, and coordinating the broader immune response. Selenium's contribution to immune strength stems from its integration into cellular proteins, which function as antioxidants, redox regulators, and structural proteins that support cellular integrity and signaling. One of the most important of these is glutathione peroxidase, which protects immune cells from oxidative damage during times of high stress, such as during an infection or inflammatory response. Without adequate selenium, immune cells become vulnerable to reactive oxygen species produced both by pathogens and by the body's own defensive mechanisms during an immune response, particularly a viral infection. The body increases its production of free radicals as a defense mechanism, which can simultaneously damage host tissues if left unchecked. Selenium-dependent enzymes mitigate this risk by neutralizing these radicals, preserving the function of immune cells and preventing collateral tissue damage. This balancing act between immune aggression and control is essential to avoid excessive inflammation that can lead to chronic diseases or worsen the severity of acute infection. Studies have shown that selenium deficiency impairs the ability of T cells to proliferate and reduces the cytotoxicity of natural killer cells, leading to a less effective immune response. Additionally, selenium-deficient individuals often exhibit increased expression of inflammatory cytokines, indicating a tilt toward an unregulated and potentially damaging immune activation profile. One of the most striking examples of selenium's role in immune protection is seen in the context of viral mutation and virulence. Research has demonstrated that in selenium-deficient hosts, certain viruses such as the Coxsackie virus and influenza can mutate more rapidly, becoming more aggressive and pathogenic. This occurs because the oxidative environment within selenium-deficient hosts allows for faster viral replication and error-prone replication, leading to more dangerous strains. In other words, a lack of selenium not only weakens the immune response but may actually drive the evolution of more harmful viral forms. This has significant implications for public health, particularly in populations with widespread micronutrient deficiencies. Selenium also plays a crucial role in modulating chronic immune-related conditions, including autoimmune diseases. Its ability to regulate redox status inside immune cells helps prevent the misfiring of immune responses that target the body's own tissues. In autoimmune thyroiditis, such as Hashimoto's disease, selenium supplementation has been associated with reductions in thyroid peroxidase antibodies and improved thyroid function. This immunomodulatory effect is not limited to the thyroid. Conditions such as rheumatoid arthritis, lupus, and even inflammatory bowel disease may also be influenced by selenium status due to its systemic impact on inflammation and immune cell behavior. The influence of selenium on adaptive immunity also extends to vaccine responses. Adequate selenium levels have been associated with improved antibody production and better protection following vaccination. This is particularly important for elderly individuals and those with compromised immune systems who may not mount strong responses to immunization. By ensuring selenium sufficiency, these individuals may experience enhanced immunogenicity and more robust long-term protection. This highlights selenium's role not only in fighting infections, but also in preparing the body to resist them through more effective immunological memory. In addition to its direct effects on immune cells, selenium contributes to the structural integrity of epithelial barriers, including the lining of the respiratory and gastrointestinal tracts. These barriers represent the body's first line of defense against pathogens. Selenium supports the repair and maintenance of these tissues by reducing oxidative damage and promoting healthy cell turnover. In the lungs, this is particularly critical during respiratory infections, where inflammation can cause tissue breakdown and increase susceptibility to secondary bacterial infections. Adequate selenium status supports faster resolution of inflammation and better tissue recovery, reducing the risk of complications and chronic lung damage. Emerging research also suggests that selenium influences the microbiome, which plays an increasingly recognized role in immune regulation. Selenium may help maintain the balance of beneficial gut bacteria, indirectly supporting immune function by fostering a more anti-inflammatory environment in the gut. Since the majority of immune cells reside in or near the gastrointestinal tract, a healthy gut immune axis is critical for systemic immune competence. Selenium's role in preserving gut integrity and microbial balance further reinforces its value as a multifaceted immunonutrient. Brazil nuts, as a concentrated natural source of selenium, offer a simple yet powerful way to enhance immune resilience. Unlike synthetic supplements, the selenium in Brazil nuts is delivered in a food matrix that may offer better bioavailability and fewer risks of overdose when consumed in moderation. The consistency of daily intake, rather than sporadic high doses, is key to supporting steady immune function over time. This supports the concept that immune strength is not built overnight but maintained through regular nourishment and micronutrient adequacy. Selenium's ongoing role in balancing immunity, reducing infection severity, and preserving tissue function highlights its indispensability in a world where immune challenges are becoming more frequent and complex. Selenium plays a critical role in protecting the brain from oxidative stress, a process that contributes to neurodegeneration, cognitive decline, and mood disorders. The brain consumes a disproportionately high amount of oxygen compared to other organs and has an abundance of polyunsaturated fats that are particularly vulnerable to oxidation. At the same time, the brain has limited antioxidant capacity, making it one of the most susceptible organs to oxidative injury. Selenium, through its incorporation into cellular proteins like glutathione peroxidase and thyrodoxine reductase, serves as a frontline defense against the buildup of reactive oxygen species in neural tissue. These enzymes help neutralize hydrogen peroxide and lipid peroxides, preserving the integrity of cell membranes, synapses, and mitochondrial function within neurons. Neurons are highly metabolically active cells that rely on healthy mitochondria to produce the energy required for neurotransmission, synaptic plasticity, and maintenance of cellular structure. Oxidative stress disrupts mitochondrial function, leading to a decline in ATP production and the release of pro-apoptotic factors that trigger cell death. Selenium helps protect mitochondrial membranes and supports the recycling of antioxidants like vitamin C and vitamin E, which further reduces oxidative burden in neural tissues. This mitochondrial preservation is vital for sustaining cognitive processes such as attention, memory formation, and learning, especially as the brain ages and becomes more vulnerable to metabolic inefficiencies. Cognitive decline and diseases such as Alzheimer's and Parkinson's have been closely linked to increased oxidative damage and chronic neuroinflammation. Post-mortem studies of patients with Alzheimer's disease often reveal elevated levels of lipid peroxidation and reduced activity of antioxidant enzymes, including those that require selenium. Low selenium levels have been correlated with worse cognitive performance in epidemiological studies, particularly in elderly populations. Individuals with higher selenium status tend to perform better on memory and language tests and experience a slower rate of cognitive decline. This suggests that selenium's protective role extends beyond acute stress responses and contributes to the long-term maintenance of cognitive health. In addition to protecting neurons from oxidative damage, selenium supports neurotransmitter balance, which is essential for mood regulation and emotional resilience. Dopamine, serotonin, and norepinephrine are all influenced by oxidative and inflammatory states in the brain. When oxidative stress becomes chronic, it disrupts the synthesis and receptor sensitivity of these neurotransmitters, leading to symptoms of anxiety, depression, and cognitive fog. Selenium's antioxidant properties help stabilize the environment in which these neurotransmitters operate, supporting healthy mood and mental clarity. Clinical studies have shown that individuals with low selenium levels are more likely to experience depressive symptoms, irritability, and fatigue. In one notable study, participants who consumed just one Brazil nut per day for 12 weeks experienced significant improvements in mood and reductions in anxiety. This change occurred without the use of pharmaceuticals and was attributed to the normalization of selenium levels and the associated increase in glutathione peroxidase activity. The participants did not change their diets or lifestyles otherwise, which points to the remarkable effect of correcting a single micronutrient deficiency. This study highlights the possibility that mental health symptoms, sometimes attributed to complex causes, may be influenced in part by subtle nutritional imbalances, particularly involving trace minerals like selenium. Selenium's neuroprotective functions are not limited to older adults. In developing children and young adults, adequate selenium is critical for proper brain development and neurological function. Animal studies have demonstrated that selenium deficiency during key developmental windows can result in lasting impairments in memory, learning, and motor coordination. This underscores the importance of ensuring sufficient selenium intake across the entire lifespan, not only to prevent decline but also to support optimal brain growth and function. Another emerging area of research involves selenium's interaction with the blood-brain barrier, a selectively permeable membrane that protects the brain from pathogens and toxins while allowing essential nutrients to pass through. Selenium appears to play a role in maintaining the integrity of this barrier by supporting the function of endothelial cells and reducing inflammation that might otherwise compromise its structure. A weakened blood-brain barrier is a feature of many neurological disorders, including multiple sclerosis and Alzheimer's, and selenium's ability to preserve its integrity may offer an additional layer of protection beyond individual brain cells and structures. Selenium influences the broader neural networks that govern behavior, memory, and cognition. Functional MRI studies have shown that oxidative stress can impair communication between brain regions, reducing the efficiency of neural processing and decision-making. By reducing this oxidative burden, selenium helps preserve network connectivity and signal transmission speed, both of which are crucial for maintaining high-level cognitive function. This is especially important for individuals under chronic stress, which increases oxidative stress and can impair brain function even in the absence of structural disease. Brazil nuts offer a natural, food-based solution to boost selenium levels without the risks associated with synthetic supplementation. Just one nut contains enough selenium to meet or exceed the daily requirement for most adults, providing a simple way to enhance neurological protection through diet. Because selenium in Brazil nuts is bound to amino acids, it is readily absorbed and utilized by the body, making it a reliable source for supporting brain health over time. This aligns with a growing body of evidence that nutrition plays a foundational role in neurological resilience, and that even small dietary changes can yield meaningful improvements in cognitive function and emotional well-being. Selenium plays a vital role in DNA protection and cellular repair system, making it a key player in cancer prevention and long-term cellular health. One of the primary ways selenium contributes to DNA integrity is through its support of antioxidant enzymes that reduce oxidative damage to genetic material. Every day, cells are exposed to oxidative stress from environmental toxins, inflammation, metabolic processes, and even normal cellular respiration. This oxidative stress leads to the formation of free radicals, which can damage DNA strands and increase the likelihood of mutations during cell division. Selenium, as an integral component of glutathione peroxidase and other cellular proteins, helps neutralize these free radicals and prevent them from causing harm to DNA. Maintaining DNA stability is critical because when mutations accumulate, they can disrupt cellular function and potentially initiate the process of tumor formation. Another important mechanism by which selenium supports cellular health is through its influence on apoptosis, or programmed cell death. Apoptosis is the body's natural way of removing damaged or potentially cancerous cells before they can proliferate uncontrollably. In healthy tissues, apoptosis acts as a protective filter, identifying cells with abnormal DNA or malfunctioning mitochondria and directing them towards safe elimination. Selenium enhances this process by modulating the expression of genes and proteins involved in apoptosis, including p53 and caspases. By supporting effective apoptosis, selenium helps the body eliminate cells that might otherwise contribute to cancer development. This is especially important in tissues with high turnover rates, such as the skin, gastrointestinal tract, and immune system, where constant cell division creates more opportunities for errors to occur. Several studies have linked low selenium levels with an increased risk of various cancers, including prostate, lung, colorectal, and liver cancer. One of the most prominent trials in this area, the Nutritional Prevention of Cancers (NPC) study, found that selenium supplementation significantly reduced the incidence of prostate, colon, and lung cancers in individuals with initially low selenium status. The underlying reason may be selenium's dual action of reducing DNA damage and supporting immune surveillance mechanisms that detect and remove early cancerous cells. Additionally, selenium influences angiogenesis, the formation of new blood vessels, which tumors rely on to grow and spread. By modulating angiogenic signaling pathways, selenium may restrict a tumor's ability to create its own blood supply, thereby limiting its growth potential. Selenium also supports enzymes that participate in DNA repair processes, such as those responsible for base excision repair and nucleotide excision repair. These systems are crucial for correcting single-strand breaks and removing damaged DNA bases that could otherwise result in mutations. Cellular proteins contribute to a redox environment that allows repair enzymes to function optimally, ensuring the fidelity of DNA replication and transcription. Without sufficient selenium, these repair mechanisms can be impaired, increasing genomic instability and raising the risk of carcinogenesis. In this way, selenium acts not just as a preventive agent, but as a continual safeguard that preserves genomic accuracy with each cycle of cell division. Furthermore, selenium plays a role in regulating cellular proliferation. It influences signaling pathways such as the mitogen-activated protein kinase (MAPK) pathway and the nuclear factor kappa B (FB) pathway, both of which are involved in cell growth, inflammation, and immune responses. Dysregulation of these pathways is a hallmark of cancer, as they promote unchecked cellular proliferation and resistance to apoptosis. By modulating these pathways, selenium helps maintain a balance between growth and inhibition, preventing the overactivation of processes that could lead to tumor formation. In addition to its cellular-level effects, selenium has a broader role in supporting immune surveillance, the body's ability to detect and eliminate abnormal or cancerous cells. Natural killer NK cells and cytotoxic T cells are critical components of this defense system, and their activity is closely tied to selenium status. Selenium enhances the ability of these immune cells to recognize and destroy cells exhibiting abnormal antigens or signs of mutation. This relationship between selenium and immune competency further reinforces its role in early cancer defense and tumor suppression. Brazil nuts, due to their exceptionally high selenium content, offer a natural way to support these protective mechanisms. By providing organic forms of selenium such as selenomethianine, Brazil nuts contribute to the incorporation of selenium into selenoproteins that perform these critical functions. Studies have shown that regular consumption of Brazil nuts can raise plasma selenium levels and enhance the activity of antioxidant enzymes within weeks. This translates into greater DNA protection, improved immune function, and more efficient removal of potentially malignant cells before they can form tumors. Selenium's role in hormone-sensitive cancers such as prostate and breast cancer has also drawn attention. Hormonal fluctuations can influence oxidative stress and cellular proliferation in hormone-sensitive tissues, creating an environment more prone to mutation and growth disturbances. Selenium helps to counteract this through its antioxidant effects and ability to regulate hormone metabolism, especially in conjunction with other nutrients such as vitamin E and zinc. These synergistic interactions highlight how selenium operates within a network of micronutrients that collectively support anti-cancer defenses and hormonal balance. In the context of chronic inflammation, which is a known risk factor for cancer development, selenium plays a key role in reducing inflammatory cytokines and blocking signaling pathways that promote cellular transformation. Chronic inflammation creates a microenvironment rich in growth factors, free radicals, and inflammatory mediators, all of which contribute to the development of cancer. By reducing inflammation at the cellular level, selenium disrupts this environment and makes it less conducive to malignant transformation. This multifaceted approach, reducing DNA damage, enhancing repair, promoting apoptosis, regulating immune responses, and decreasing inflammation, positions selenium as a crucial nutrient in the prevention and control of cancer-related processes throughout the body.